Good afternoon, everybody. It's already afternoon in Ontario, and thank you, Stu, for the introduction. I'm trying to share the screen. Okay, there we go. So I'm greeting you from this beautiful University of Guelph campus. I've been here since 1998, and I came here to do my graduate studies and then remained as virologist at the animal health laboratory. So pretty much last 23 years or so I spent in this kind of area on a campus. And over those years, I've seen a lot of change. I mean, the campus itself has changed some parts of the campus dramatically that if you weren't here for quite a while, you wouldn't recognize it. So Animal Health Lab is located right here. A number of years ago, we moved to this new building, and we use that as an opportunity to change processes and to reorganize and to merge labs and to use new techniques. So this change has been happening very much for those 20-something years that I've been around. From my window, I have this beautiful view of Johnston Green that now has got like those big trees, and the only thing that I know about it is that it's going to keep on changing. Soon enough, it's going to look like fall, and then again, well, this is southern Ontario, but we do get a lot of snow and can get pretty nasty here. So one thing that's that has happened over the years is that we've never been bored. And I like to show this cartoon, which one of the students in my lab put on a whiteboard and said, nothing to do, there's always something to do. And we have learned through those years that what we need to do is to again adjust the processes and change the way we do things to be prepared. And I like to quote Louis Pasteur here, who once said that chance favors only the prepared mind. And so we've been trying to do what we can do to become prepared. And as Stu mentioned, for me personally, coincidentally, and today we talked about adenoviruses, I've been, getting ready since, the last century. It was in 1994 that I first started working with adenoviruses, and I did my master's with bovine adenoviruses. And in 2001, I finished my PhD using fowl adenoviruses as what I was working with. So, cut long story short, there's lots of history between me and adenoviruses, and the rest of my presentation, I would like to actually start with the history about fowl adenoviruses. I'll talk about the virus itself, I'll talk about what we do in the lab, and also I will tell you what is the current situation, at least from my lab's perspective. One interesting and less known fact about fowl adenoviruses is that the first isolation or detection of the virus was unintentional. They actually tried to make agent of bovine lumpy disease in chicken eggs and isolated the virus to detect if there was something there, and they thought that basically that was the agent of lumpy skin disease, but it was actually an adenovirus, and they called it, because they didn't know what to call it, they called it chicken embryo lethal orphan, which is virus, which is what we call now fowl adenovirus type 1. And first intentional isolation of adenoviruses occurred in just 1 year later, and was actually from quail and from respiratory disease in quail. The virus, that's the same virus, can cause pretty nasty disease in quail. In terms of inclusion by hepatitis, which is the title of today's talk, and the first report that I could find goes back, even before the time I was born, was 1963, where they actually found internuclear inclusion virus in liver of chickens that had clinically hepatitis, and they were dying of hepatitis, but they didn't know what was causing it at that time. So only maybe 10 years later, there were several groups which found adenoviruses in samples from birds that had included bad hepatitis, and those groups had found a whole bunch of different adenoviruses, and they were called 1, 2, 3, 4 And this classification scheme was basically complicated by the fact that clearly those viruses were really spread throughout the world, and there was different groups in Japan, Northern Ireland, US, Hungary that were working, and you can see the references. This is all from the 60s and the 70s. So the way that the numbering and calling different strains, different numbers worked was inconsistent in the past. So you can see that some of the viruses were called 8, 3, or 5, and the other ones were called 3 or 6. Some were called 9, 8, 5, or 10. Cut long story short, just forget about all of this. Now we're using this ICTV classification, which stands for International Committee for Taxonomy of Viruses. And fowl adenoviruses are now divided in 5 species, and pretty much the ones that we're seeing that we're having an issue is the ones in species E, which are adenovirus types 8 and 8A and 8B, and then primarily from group D is 11, and from some other countries actually is type 4. So just to show you a cartoon of an adenovirus and an electron micrograph of an adenovirus, they're generally speaking medium-sized viruses that don't have an envelope, so they're pretty sturdy. They have this typical icosahedral sheet with those projections at the end, which are called fiber protein, which virus uses to attach to target cells, and they have double-stranded DNA as their genomes. So generally speaking, why would we care? Well, there's so many viruses that we don't know exist, but clearly these viruses, scientists and practitioners and industry all over the world care for quite a long time, because they do have worldwide distribution. Even just take a look at those previous slides that I showed that, in the 50s, 60s, scientists in many countries already worked on them. So initially, this first report was about pneumonia, and certainly some of the adenos can be associated with disease of respiratory tract. Over the years, they've been also implicated in various conditions such as gizzard erosions, provincialitis. Certainly, they can cause pancreatitis. IBH-like virus will cause severe pancreatitis. They were involved even with suppression, but pretty much most reports in recent years related to so-called hepatitis, hydropericardium syndrome, and inclusion bladder hepatitis. And the difference between these two ones are that IBH, or inclusion bladder hepatitis, is something that we've seen in Canada, US, and typically Americas and in Europe. And typically virus strains that we now call type 2, 8, and 11 were involved, whereas hepatitis, hydropericardium syndrome is serious disease, can be quite dramatic, but was never really described in Canada and US, or not even in Europe, but in Asia, in Mexico, in Central South America. So for some reason, the distribution or application of fowl virus 4 in this HHA syndrome, is still kind of unusual. In terms of epidemiology, the most severe outbreaks occur when the virus is vertically transmitted from hens to eggs to the progeny. And many years ago, we had them as early as, cases as early as 6 days, with mortalities of 35, 40, 50% in those kind of situations. But, that can occur, like vertical transmission can occur in all the breeder flocks because adenoviruses can cause latent infection and can get reactivated. And again, could be, that there's a new virus that was introduced later in the life of the breeder flock. Could be immunosuppression as well, or could be a combination of all the factors that I listed above. So for zoonotic transmission, typically, there's different scenarios as well. Could be that because of mixing of broilers from various breeder flocks, some of those ones are from antibody negative flocks, and they basically get exposed when they get placed. It could be a combination of mixing broilers that are shedding or not, and immunosuppression, and again, various factors. So to cut long story short about epidemiology, the way that virus came and caused a disease is sometimes not really clear. In terms of what we do in a lab, well, historically and typically, just post-mortem examination for experienced practitioners is good enough to determine that most likely this is IVH involved. In lab, we do histologic examination. In older days, we do virus isolation, which would then follow with electron microscopy, at least in my lab, to confirm that was adeno involved. Nowadays, we do PCR, and viruses historically in old days would be characterized by serotyping, but that's typically not done anymore because very few labs have access to all the reference here that you need to do serotyping. But rather we rely on genotyping, basically looking at genetic material of the virus to determine the strain of virus that we're dealing with. So just to show you a few pictures, the slide A shows typical appearance of the liver in birds with inclusion body hepatitis, and here these hemorrhagic lesions are actually in pancreas, which can be quite dramatic. When you look at the histology, again on the left side, the slide C shows the liver cells that are infected with lots of inclusion bodies in the nuclei of the cells, and the one on the right of the slide D shows the pancreatic cell infection with adenovirus. This is appearance of the cells before here and after 24 hours after inoculation. And foul adenos typically grow really well in cells. It's pretty easy to isolate them, can be grown to high titers. So basically, when you put a sample that contains virus on cells, so if you look at this, looks like a kind of nice lawn, and then 24 hours you have grub infestation, and your lawn is, or it looks like it was after drought. So it's very characteristic, but still not 100% sure that when you have this kind of appearance of infected cells that it's foul adenovirus, because there could still be some other options. So typically in old days we do electron microscopy to confirm that in cell culture supernatants we see those icosahedral shaped particles, which confirm that that was adenovirus that caused cytopathic effect in cells. And if you were to do transmission electron microscopy, basically slice the tissue to see what's in those cells, you'd see that how packed those nuclei of the cells can be. Those dark dots are all adenos. This on the left is again a liver cell, and here this is a pancreatic cell. So in livers, and when we do PCR now, when we can semi-quantify, sometimes we get really massive amounts of virus, like crossing point values that we get are super low, suggesting that the load of virus in the real tissue is super high. So PCR is what we've been doing now for over 10, 15 years. Is primarily the almost completely abandoned virus isolation for a number of reasons. But PCR gives us also an idea of again, the load of the virus in the sample that we test. And also the primary advantage of PCR is speed, because we solidify those processes and electronize them, so to say. So there is as little as possible human involvement to minimize any chance of errors occurring. And in my experience, lab errors are typically clerical errors. So relying on electronic interfaces and instrument machine called results improves the confidence in results that we produce. So in terms of virus characterization, as I mentioned, serotyping is barely ever used anymore. Not even I have all reference sera. I have some. And as virus change, you would have to produce your own, and inoculation of birds to produce sera is not routinely done anymore. It just gets very complicated. So rely on genotyping. As routine genotyping test, we use comparison of sequences of hexon gene, and more recently we also did some whole genome sequencing, but in other labs all over the world, there's been more and more reports of whole genome sequencing. What's the difference? The difference is that With the hexon protein gene, we sequence only this blue protein that you can see here. This is the most abundant protein of viral particles, but it is not the only one that's involved in antigenic properties of the virus. It's not the only one that can predict pathogenicity or virulence to the virus and so on. For example, fiber is super important because even minute change here will make the virus change significantly. So that's why whole genome sequencing long-term is going to become pretty much standard because it will give you much more precise and much more in-depth view about what you're dealing with. So when I finished my graduate studies and started working at the Omaha lab, again, I was prepared to work with adenovirus as much as one could be. But the thing was that at the time, there was not much interest in adenos, right? It was the time where basically adenos or inclusion-bound hepatitis was a sporadic disease. And if you look at this chart that shows the numbers from 2000 to 2006, you'll see that between 2000 and 2002, 3, we had maybe 20, 30, 40 cases a year. And again, they weren't dramatic. There was knowledge that the viruses are there, but it's just a little increased mortality, a little increased condemnations, and no significant impact on production. However, all that changed as everything else changes in life, as I mentioned, in 2004, because suddenly from having 20, 30 our numbers in terms of detection of cases, which is just the tip of the iceberg because that's only based on samples that we tested in the lab, went up like 5 or 6 fold. And this high number of cases continued throughout the next few years. And this is where we started doing genotyping to see what has happened here, what is it suddenly changing, and what it appears to have changed that we had introduction of new strains of fowl adenoviruses. In particular, if you look at this chart that summarizes the numbers of genotypes, here out of 246 cases that we genotyped over those few years, roughly 40% of them type 11, and then there was various types of adeno 8A, that again together presented roughly 40%, and then 8B and 2 were the last 20%. So there was kind of 40, 40, 20 a kind of division between genotypes that were associated with those cases. In particular, some of those type 8 didn't appear to be present in Canada before, and type 11 certainly was present in much, much lower level. So then somewhere about that time, we still were doing virus isolation. We started doing PCR, so PCR became the predominant test. And I just show you this chart because, again, you need to be careful how you interpret your results. Here it would appear that this increase in PCR testing occurred in 2010-11, and that we had not outbreak, but that's not really the case because that was a research project that generated artificially high numbers for those 2 years. So in this slide, I'm actually removing those cases just to show you, real life, not research. Again, research was good for another purpose. I'll talk about it later, but this is how numbers look like over the years since 2008. So that's like 14 years now. For 2021, I included first 5 months, so it's up until June 1st, so not even 6 months. But you can see that basically numbers were on the downswing up until 2016, and they went up in 2016 and now are high. And actually 21 is probably going to be much higher because these numbers are only for first 5 months. So something has changed here again, that there's been massive amount of change in terms of these numbers. And before I talk about it, I just want to show you that again, we are Ontario-centric lab, and most of samples we do get here are from Ontario. So roughly 57% of samples we tested were from Ontario. But in particular, since 2016 onward, we started getting more and more samples from Alberta, from British Columbia, from the United States. Actually, we get lots of samples. So roughly even 10% of samples that we've done were from the US. So we kind of get an idea of what's happening all over Canada and US. In 2012, we even got 4 samples from Mexico, but it's kind of harder to get import permits. It's simple for us to get samples from the US. We have import permit for a whole bunch of samples for types and tests from the United States, but to get them from other countries, it kind of is often harder. In terms of commodities, this includes pretty much all of them, except for that research project. But we've done some surveillance project in other commodities, so most samples are from broilers, but we did look into broiler breeders, and in 2015, 16, and 17, there was surveillance in small flocks. We get occasional, exotic birds, and we had also some samples from turkeys. And over the years, we did have 2, I believe, positive samples from turkeys that we genotype later. In terms of positivity, pretty much it varies from year to year. And if you look at maybe last few years, as we get more and more, so the percentage of PCR positives is getting higher. So almost 100% of samples that we received this year ended up being positive. Basically, overall, if you look at all of them, almost 90% of samples that we received from broilers, with suspicion of IBH were positive for at least one adeno. Again, that goes back to the experienced practitioner can be pretty confident in results, but probably 1 of 10 are those ones that are not necessarily your typical cases. So then doing additional lab testing so with that. And in terms of genotyping that we've done since 2008, again, the numbers jump here in 2010 and 2011, and when I remove those ones, again, genotyping pattern follows the same trend from going slightly higher 2010 and then dropping down to almost nothing. So you see in 2015 we did genotype only 6 samples, and then going up since 2016, and this year already in the first 5 months we did almost as many as we did whole last year. So again, my prediction will be that this year's numbers will go up significantly higher than what we had in 2020. In terms of commodity groups, over the years we've done again, primarily broiler samples, but I'll do talk a little about what we found in broiler breeders and small flocks. I'll give you just an example of what happened during that research project. So I think it's going to be very informative from that perspective. And we had 2 samples from turkeys that we successfully genotyped. So by the province, again, as you can see, US is even more represented here. Roughly 12% of samples were from the US, and from the West, again, another 12. So Ontario is still almost between 50 and 60%. But in more recent years, you can see this increase in samples that arrive from Western Canada and from the US, whereas Ontario is more or less steady, goes a little bit up and down, but there's not that much increase in what we get here. So I'm showing you this table just to show you the diversity. I'm not going to talk about results based on this because it's overloaded, because basically this is summary submissions of all types from all provinces, from all commodities. And if you look at the types of viruses that we genotype here, we have 1, 2, 3, 4, 5, 6, 8, A and B, 9, 10, 11. So the only one that we don't have here is the 7, and we have this turkey isolate as well. So to make this a little more understandable, I took out, for example, this first part is that research project that I talked about that was done in 29, 10, 11. So what was done here, we collected samples from 231 broiler flocks at processing and then tested them for a whole bunch of other diseases and viruses and bacteria. But basically, when you look at fowl adeno results, and those are not necessarily birds with problems. Those were just random sample. That's roughly 20% of all birds that were, or all flocks that were processed at the time in Ontario. So basically, at processing, 215 samples or 215 flocks were still shedding, were positive for adeno by PCR, and 151 already seroconverted to fowl adeno. So when you put this and this together, because some of those negative flocks were serologically positive and the other way around, it ended up that 96% of those flocks were positive for adeno at the time of processing. And when we looked at all different genotypes of adenos that we detected in those flocks, again, those are not necessarily related to IBH at all. It's just, surveillance, what's present in those flocks at the time of processing. The most common one is this fowl adeno 1 or SILA virus. And that one was present in over 60% of the cases that we detected. Now, the second one is not surprisingly is 11 because we know that that one is involved in IVH. And also there was some or several different subtypes, 8 and 8B viruses. One of the surprises that we did notice here was this type 4. There actually was one flock that had type 4, which was known to cause serious issues in Asia and South America and some parts of Africa. So the other part of surveillance was also in broiler breeders. So we looked at broiler breeder flocks, and some of them had issues with IBH, or the project had issues, and some were normal flocks with no history of IBH in their progeny. And again, SILA was the most commonly detected virus, this type 1. And surprisingly, again, we got 6 out of 29 flocks were shedding type 4. Again, that's the one that's involved in HHS or hepatitis hydropericardial syndrome in those other countries. And finally, this is the slides from surveillance in small flocks. And basically, here there's another bit of a surprise. Wasn't that anymore because now we knew that we had this virus. So again, type 4 was the most commonly detected virus here in those 2 years of the study. It was a huge number of flocks that we did, and these ones were not with IBH. Again, those are just backyard flock with some kind of issue, and lots of backyard flocks have some kind of issue. And interesting enough, SILA wasn't the most common one, but rather it was again type 4. And the other thing is that there's great diversity here in those backyard flocks: 1, 2, 3, 4, 8, 9, and 11. They're all present there at really low levels. Now, that is definitely different than what happens in broilers, because this table contains the numbers that were from broilers that were submitted because they had IBH. And here I included only the numbers since 2016 because when that change has occurred. And again, interesting enough, not a single case, and actually not a single case ever we had with fowl adenovirus 4 in broilers that had IBH. All of those ones were all incidental findings in backyard flocks, in broilers at processing, or broiler breeders. But clearly, this virus is not associated with IBH in at least not in Ontario and US based on samples that we received from the US. So what has changed here suddenly is that there is no more 40-40-20 split. Remember that first outbreak that was in 2004, 5, 6. Those years we had 40% type 11 virus, 40% type 8A, and 20% the rest. Now this year, or so since 2016, actually the type 11 numbers fell down to only 60%, type 8 dropped down to barely 1.6%, and over 80% of samples that we genotype from Canada and from US are type B. In particular, 2 of the strains of this type B that I'll just talk a little more detail in a few minutes. And this is what happened with 8, again, from 40% before, and even 2018 was still at 30%. It basically dropped down to the point of barely 3% of samples that we typed this year were fowl adenovirus 8, while the numbers for those 2 strains of type 8B have been increasing steadily. And again, there's 2 kind of subtypes. They are pretty close related, but I can clearly differentiate between those 2 ones, so I keep them separate. So there's this one that we first detected in 2016, and the other one was in 2018. And I said 2016, yes, 2016 in samples in Ontario. So initially, remember that in 2014-15, we had genotype very small number of samples. But when I look back, when we started getting issue with this new A to B subtype virus in Ontario, I look back in my older genotyping data, and actually we did detect identical virus in 2014 in samples that we received Nova Scotia. And then in 2016, we started getting them from Alberta, British Columbia, US, and Ontario. So clearly this virus arrived to Canada and probably some other parts, much earlier than we thought, but may have been due to the way that the industry works, may have been confined to just some parts of Canada or US or some other countries before it spread worldwide. And I'm saying worldwide. Why? Because if you look at this is the summary of hits, the highest hits. That's if I look at the sequence of the virus and I compare it with other viruses which are in GeneBank, which is international depository of virus sequences and bacterial sequences, other sequences as well. But anyways, you'll see that the best matches to this strain were from Japan, from China, from Peru, and there's no matches from US. Well, again, we know that it was ready in US in 2016, but nobody deposited this gene bank. So you need to take some consideration about that. Not necessarily everything is going to be in gene bank is going to take a year or 2 or 3 before scientists deposit those sequences there. And clearly, this suggests that even a few years ago, because this came from 2013, right? So in 2013, this kind of virus was already in Japan. And we know that was 2014 was already in Nova Scotia and in Ontario in 2016. So anyways, this depicts the worldwide distribution of this new type A to B that has been causing really serious problems in many parts of Canada and clearly in the world. If you look at this other one, that again, we first detected in 2018, that one, again, there was already virus like this in the US in 2016. So 2 years before we had it here for some reason. So this virus is also quite similar or 100% identical to viruses that were found in Turkey and Indonesia, and quite similar to some that we found in this first outbreak. It was not common in this first outbreak, and it's not identical to it, but certainly it appears to be quite related to this A to B virus from the outbreak in Ontario in 2004 and 6. And actually, this one is from British Columbia because it says BC right here. That's how I call it. And question now is, so what's happened? Like, why did this happen? And does anybody know what will happen? I actually don't. I'm not pretending I do, and nobody can give a full explanation, but we can just look at a few things that may have played a role in development of this kind of situation. And it's related to, again, the structure of the virus. And remember, I said that in terms of antigenic properties, the antigenic kind of a thing is the sum of this fiber protein, hexone protein, pentone protein, and pentose base protein. So basically, these would probably put all together represent 90% of antigenicity property of the virus. And what has been just recently discovered, which was in 2019, that basically the foul adenoviruses can recombine. And I know from my lab experience that we often have mixed infections because we can see sometimes that the sample contains, well, one major virus, and then there's something else that's clearly adeno, but we can't clearly read it because the way that technology works. But cut a long story short, when the whole genomes from 27 isolates of foul adenovirus were well genotyped and their sequence is determined, then scientists, and this study is from Europe, from Austria, but actually some viruses are from Japan. So again, that sounds like even when this was done, included viruses from several countries. So cut a long story short, these viruses can exchange genes for those proteins, or half of the virus can be from one subtypes and the other half virus can be from another subtype. So that when you generate hybrid viruses, which again, just next year was also confirmed in a study from China, where they basically found that there was viruses that contain, fiber gene from type 8A and the rest of it from type B. So again, at least 2 studies that were recently published that show that the change is constant, if nothing else. And basically the change generates those new viruses that somehow will then end up being spread all over the world and cause serious issues in all countries that have some industrialized poultry production. So this kind of explains also about some issues that if you read papers and some studies and even my lab, how do you interpret cross-reactivity versus cross-protection? And clearly we can't. I mean, you could if you had really focused on that. It's doable, but no lab nowadays, at least to the best of my knowledge, has the ability to do that. In North America, again, there's this good lab in Austria that does lots of work. They may do some stuff, but this can actually well, recombination and generation of new mutant viruses sometimes works to advantage, because in this example, fall 8A vaccine protect against 8B and 11. And the reason probably for it is they didn't know it, because this was published before those other studies that show this recombination occurs, that often they basically, it's probably, that recombined virus that had pieces from several viruses generated those kind of serological results. So this protection, you could almost have universal, monovalent vaccine if you pick the right recombinant, but at present nobody really knows how to do it. And to cut a long story short, and to bring it to the end of my presentation, the only thing that I've, so to say, learned over the years, that the only thing that's constant is the change. And in this example of its inclusion by hepatitis and following the virus infection, clearly is that this 8B or 8B strains have emerged worldwide, and since 2016, they've become the primary cause of IVH in North America, approaching or exceeding 80% of the cases that we've seen. We also now know that mixed infection is quite common, and that recombination can occur in field conditions to generate new hybrid viruses, and we can consistently predict cross protection and cross reactive using the current methods. As a control, still, natural exposure, is one way that one can obtain it, and the other is vaccination. And unfortunately, in North America, we don't have, as far as I know, licensed live vaccines, which would be easy to use and inexpensive, but we gotta rely on autogenous kill vaccines. And basically, that's the end of my presentation. Before I finish, I just like to thank everybody in my lab for their hard work and contributions. And this picture is, clearly pre-COVID. So as soon as we don't have to wear a mask anymore, we'll take another picture. So maybe next time, if I talk at BC Poultry Symposium about something else, I'll have updated picture. But for time being, I really want to thank my team, because I do have a great team. We have lots of equipment, good lab, and we do lots of work that hopefully serves a good purpose. And finally, I want to thank you for your attention.
Thank you so much, Davor. We'll now ask Dr. Gigi Lin to take the stage, and from there we will entertain questions if we have time. And obviously, we work to post this information on our website at a later date. So we will now transfer to Dr. Lin. Thank you.
Okay. All Great. Move something around. Perfect. Great. Well, thank you very much. Thank you, Dr. Ojkic, for that really in-depth presentation about the virus, the adenovirus itself, a little bit of its history and a trend, how it changed in the last couple of years. And for the next 30 to 40 minutes, I would like to take the opportunity to focus more on farm control strategies, so information that are more relevant to grower and the farm managers in terms of IBH control. And when I talk about disease challenges, I always like to talk with this really classic epidemiologic triangle, which is a basic model to understand how disease problems and how health problems evolve, and to understand and to appreciate that it's very complex and there's a very dynamic interactions among them that can involve the disease outcome. In this case, we have our disease, inclusion body hepatitis. And for it to happen, of course, we need the virus, which Dr. Davor has talked a lot about. And then the next thing we need is our victim, the broiler chickens in this setting. And the virus and the broilers, they're commingling in the same environment, and how the environment can also impact the outcome, depending a lot of many factors that are involved. I'm going to pick the three most prominent factors that are associated with IBH outbreak. I often get the questions from growers, also from farm managers, from our clients, why do I get IBH from this flock and not another? Why is it always on a particular farm, or why is IBH always on a particular floor? The expressions of IBH is very complex, and it evolves around the three main factors that I'm going to identify. The first one is the maternal antibody, which is the amount of antibody and a type of the antibody that the broiler chickens inherit from their parents, the breeders. And the second one, always very important in any kind of disease prevention strategies, would be the immune system of the birds, the immunity, which means that how possible their ability to fight against a particular disease. And then also, very importantly, the amount of virus in the environment. All of the above will contribute to the expressions and the severity of the clinical signs. A flock, a particular broiler flock, will have the greatest risk and more severe presentation of IBH when, first thing is when you have an absence or a very low maternal antibody in the broiler chicks to a specific adenovirus serotype. And obviously, any kind of immunosuppressive conditions that impair or slow down the chick immune response to a particular disease. And also in a situation where you have a highly contaminated environment with avian adenovirus. I want to start with the first level of protection in terms of controlling IBH outbreak. Before I get into the farm strategies, I'm going to briefly talk about what is the level of protection and what's the work that's been established in the broiler farm and also at the hatchery level. First of all, the maternal antibodies, how the breeders get that maternal antibodies to pass on to the chicks. So as Dr. Davor mentioned in his last slide, natural infection being one, which is the birds being naturally exposed to a few IBH virus. The second means, it's a very important means of, which is vaccination. We have been talking a lot about, which is the immune system that are being artificially induced by on-farm vaccination. And the breeders, after being exposed to the virus, either by natural infection or vaccination, they will generate those tiny proteins in their body, which is called antibodies. They are used in the immune system, circulatory system, to identify and also neutralize foreign objects like bacteria and also viruses, to allow the body to fight against a particular disease challenge, in this case, IBH. And then when the breeder carry it, how it works at the next level is when they lay the egg, they will pack all the antibodies with their nutrients in the yolk, which is a significant amount of content in a broiler egg. This is how a broiler chick inherited maternal bodies inherited that first line of immune protections from their parents. And how autogenous vaccine is manufactured is when we have actual IBH cases. So for example, like there's 2 broilers that come with IBH, we will extract the liver tissues and then send it to the lab to identify and also isolate the particular virus from the most prevalent cases in a particular location. And then we send that isolated off to the vaccine company and then to make the vaccine to vaccinate the local breeders. This process is very important to be kept up to date. We're constantly looking at viruses that are new to a particular area or has been like mutated to make sure that the breeders are being vaccinated with the most up-to-date autogenous IBH vaccine. This is how a brief summary of autogenous vaccine works. This is why when I talk to my clients, I always encourage them to, even though you're very sure that you know that you have an IBH case, very obvious signs, I highly encourage people to submit the liver tissues to your veterinarian so that we can continue to work on the disease surveillance and also updating our autogenous vaccine program. Now, at the next level would be the hatchery. Remember I mentioned about how the maternal antibodies are being packed in the yolk sac. This is all comes with that tiny egg. And it's very important that if this yolk material with the nutrients and also the antibodies not being used and not being absorbed properly, you're gonna end up with the chicks that are more susceptible to infectious diseases. This is why chick quality, a good chick quality and good yolk utilization plays an important role. Looking at these 2 chicks, like from the outside, they're day-old chicks and they both weigh 42 grams. The first chick here only has 2 grams of yolk material left. Majority of them has been well absorbed in their circulator system so that they can have a more advanced like immune system, improve gut health, and have like way better ability to fight infection. Versus the chick below here, maybe these chicks have probably a yolk sac infection or a suboptimal incubation conditions. Even though the chick has the same weight, they have a really big like yolk sac, where the yolk sac is not supposed to stay this big like for the rest of the life of the chicks. It's supposed to get absorbed properly within the first couple of days of life. So in turn, if a chick is not taking all the nutrients and the immune system that are packing it, it's obviously going to be more susceptible to any kind of diseases. So good chick quality and yolk utilization does play a very beneficial role in disease prevention. Now we talk about the maternal antibodies level on the farm, on the breeder farm, and how it packed like at the hatchery level in the first couple of days of chick's life. I'm going to spend the most focus on immunosuppressions, and this is a level where growers and also farm managers can make a more important like impact on the disease outcome of IBH. I'm going to categorize the immunosuppressions factor into 2 big groups. The first one that I'm going to talk about is management and environmental stressors. This is the most common source of immunosuppressions that we have seen like on farm in field challenge associated with not only IBH cases, but also many, many other disease challenges. Going to start with a case study. This is actually a real case that I got in the practice over last year. This is, I call Farm A, which is a conventional broiler farm in the Fraser Valley in BC. It has been having recurrent IBH outbreak for 4 consecutive cycles. They're from a mixed like breeder source with under the same autogenous vaccine program. They usually break with IBH from 23 to 26 days of age, like kind of vary. You can see the mortality rates from the IBH impact wasn't horrendous. It's still not ideal, something ranging about like 2 to 6%. So we always wonder why. Why does it happen to that same farm? It has like the same breeder sources, which was placed in many different farms that didn't have an IBH history. This is a picture from one of the submissions that I took from this particular farm. And obviously this farm, all the other birds that I look at also have IBH, like the classic liver, generalized congestions, icteric lesions. And on top of that, I took this picture because it doesn't need a veterinarian or a pathologist to tell you there's something wrong, there's some red flag with this particular flock. You'll see this bird's like poor feathering, the feather conditions not great, and there was lots of like wet litter, dirty litter that were covering all over the body of this bird, like the head, the ventral abdomen here. And on this same particular flock, I also received some complaints like from the processing plant where they have a really high condemnations from polydermatitis, which is food pad lesions that we can see. So lots of these birds have a different degree of wet litter. And we know that like, from our experience and from the pathological findings, IBH is not generally a disease that will cause a very, very severely wet litter. So this makes us into, trigger us into go into further investigation in this particular farm. So a cycle after this, we went to a farm for a farm visit at 14 days old, which is a week prior to where they usually break with IBH. And at the first glance, like it was kind of difficult to find out what happened, except that I did see like some wet litter along the water lines, like with some leaky water nipple drinker. It took me a while to finally like pay attention and realize that among their feed lines, a couple of their feed pans were actually completely empty. What happened was there was some kicky feed like in the feed auger, lots of wet feed got stuck in it. And this is a little bit of older style feed pans where it makes it very difficult for an even feed distribution. When I do an actual count, like walking through the barns completely, it ended up like we found 10% of the feed pans like without feed constantly, or just like halfly full, or just 10 to 20% full. Feed interruption, it's a very big factor that can cause like an immediate like drastic impact to the bird's body and contributing to the risk factors for IBH. What we see in feed interruption, the first thing that will happen in a bird's body would be a hypoglycemia stage, which is low blood sugar. This is very common sense to understand. When we are hungry, when we are off feed, like we can actually feel that hypoglycemia state. And I want to bring out Dr. Daniel Van's research on biochemistry. Dr. Daniel Van, he is a pioneer and expert in studying blood chemistry in avian species. And blood chemistry has been a very valuable diagnostic tools that are widely used in human medicine and also small animal medicine. And it has been just started like with Dr. Van's research, like to take a closer look how the pathogenesis and like disease cases like can help us understand like disease pathogenesis in avian medicine. So what he found is in IBH cases, he found that like 3 status in the blood chemistry, including metabolic acidosis, hypoglycemia, and also hypocalcemia, which is low blood calcium, can significantly contribute to severity of IBH. I'm not going to talk a lot about metabolic acidosis and also hypoglycemia because these are concepts that are very complex, and it's more complex to understand without any medical background. So I'm more going to be focusing on what at the farm level that growers and also farm managers can help to mitigate would be hypoglycemia, low blood sugar. Why is hypoglycemia so important? Hypoglycemia is low glucose. So glucose is the basic energy source for all animals, like not only birds, and they're also the sole energy source for the bird's immune system. When you have a decreased blood glucose in the body, there's definitely not sufficient energy resources to support the immune system, then the immune system is unable to produce like sufficient antibodies and all the other immune cells to function properly to fight the virus. And consequently, you'll have an increased severity of IBH and other diseases. The analogy is almost like your car needs fuel. It's the same thing that your immune system does need that glucose source to function properly. And why the role of hypoglycemia is extremely important in IBH compared to other diseases is, remember Dr. Davor mentioned that the target organs of IBH is not only the liver, but also the pancreas, which are the two key organs that regulate the blood glucose level. So once you get an IBH, you get a hypoglycemia stage, you get IBH. It's almost like a vicious cycle that with that hypoglycemia stage is going to drive the severity of the disease. So what are the common causes of hypoglycemia on farm? I'm talking about all the management practice that can cause hypoglycemia. First of all, as we mentioned, like reduced feed intake, it's a big one, any kind of feed interruption. So make sure you refuel your feed system very closely. Sometimes it does take time to like walk through and check through every single pans, especially your chicken pans, to find out where the problem is. And also don't forget about other factors that stop the birds from eating. You can have as much feed as you have like physically available from the birds. But if the barn is cold, if there's high carbon dioxide level, like poor air quality and poor ventilation, the birds are not going to eat as much, and it will drastically affect their appetite. Any kind of situation that the birds need more energy, chilling, which is very common. I see that more often in the winter time. I see many cases with IBH that with birds that comes in, not only seeing IBH, but also ascites, which is a condition that can be predisposed by chilling and also poor ventilation, overheating, and also any kind of factors that can affect gut integrity, that affect the normal glucose absorption. For example, overheating at an early age can also drive the birds to a hypoglycemia state. Always going back to the basic with immunosuppressions and looking through environmental stressors, like going through TFLAWs, have a very good checklist that you go through every single one of them routinely, like cycle after cycle. The second part of immunosuppression would be other diseases that the broilers are exposed to. For example, like infectious bursal virus disease, chikungunya virus, and Marek disease. The two last diseases that are on the list here, chikungunya virus and also Marek disease. These two diseases are being very well controlled and with the robust like vaccination program in the BC and also particularly in the Fraser Valley. So they haven't been a main concern in the last couple of years. So I'm not going into too much detail about that. But for other areas, if you're living in other geographic location, other countries, definitely keep these two viruses like in mind as a factor, like as an underlying immunosuppressive factors. Looking at case 2, so our farm B, this is a conventional broilers with 3 double-deck barns. We current IBH for 3 cycles. This particular farm has quite a high mortality level in one cycle with up to 20% loss in one particular barn. Also saw a really high condemnation rate with lots of condemns from bacterial hepatitis. The break was particularly early compared to majority of the cases that I see. They bulk of IBH with between like 11 to 15 days of age. And one thing to know that historically, this farm has been one of the top performing farms. Like when we go on a field call, like you can hardly see any red flags like from their environment or from their management practice. On the necropsy findings, the birds were on feed, like well flashed, like with very good body condition. And we did see the classic IBH lesions. And one thing that we also saw was a very small bursal size. When I saw the smaller bursal size at first, I wasn't very alarming because IBH itself is an immunosuppressive disease too. So sometimes like you'll see as a normal necropsy findings with birds with IBH, the bursa just basically looks smaller. But in that particular case, I took some samples and submitted for histo. And quite interestingly, it came back with bursal atrophy, which is a shrinking of the bursa, and also with some suspect lesions that that makes us suspect that there's some IBD virus involved. So to follow up with this case, we did more diagnostic tests on the end of flock. So we take some blood at the end of the flock serology. So this is a report from that actual case where we find really high significant titers level against like infectious bursal disease virus. It was up to almost like 30,000 like level in this case. So very, very high antibodies against like bursal disease. So definitely a serological evidence that shows us that there were some like underlying bursal challenge in this particular case as well. We also sent the samples to Dr. Davor for virus isolation and also genome sequencing to make sure that the reaction was not from a vaccine. It is actually, in fact, like from wild field strains that they were exposed. So on top of IBH, they also had immunosuppressive like virus that were impacting them, which is infectious bursal disease virus. So IBD virus, it's basically a virus that damaged like the bursa of the birds, which is a very important organ for immune function. I always refer it to almost like a silent killer, because in the North American strain with IBD, you usually don't see very obvious signs. You usually don't see like an immediate mortality caused by infectious bursal disease virus. Whereas like in the North American cases, you see more like a very vague sign, like for example, like poor performance, like lag in weight gained, higher condemnation, higher incidence of secondary diseases. It's almost like, the analogy is almost like AIDS in human, where you won't identify like physically from the outside that seeing a person like with AIDS, but these people are definitely more susceptible to a secondary infection. And when you have IBH and IBD itself can be separated, can be an individual disease that present in a flock. But when you have IBH and IBD together in the same bird in the same flock, it can significantly increase the severity of the clinical impact. And IBD virus is what we call very ubiquitous. It's basically everywhere when you have like poultry productions, very widespread. And unfortunately, it's a virus that is highly resistant to many environmental conditions and also disinfectants. So the control really rely on robust, dynamic like vaccine program at all levels. So I encourage you guys to talk to your poultry veterinarian regarding the control and prevention of IBD. And one thing to keep in mind is the disease is very, very dynamic, and it's very important to monitor it. So it doesn't mean that for one cycle you don't have IBD challenge. It doesn't mean that the next cycle it won't come back. Like sometimes like even the titer levels can be very confusing like to read to. We talk about the maternal antibodies, talk about immunosuppressions, and now why are we still getting the IBH from the same barn if you have like a perfect like farm setting, like management practice, and also like good vaccine, robust vaccine program? The next thing is to consider the amount of virus. Even if you have the best vaccine, best immune system, if there's lots of viruses like in the environment, the IBH can still get to the birds and cause like a big problem. Because IBH is not always transmitted from the breeders. Horizontal transmissions are very common, and they have a very, very short incubation period, which means that the virus, like once it's attacked the birds, they can replicate in the birds, and the birds can start shedding the virus like within 24 to 48 hours. So when a birds like get infected, they will shed the viruses like through all the bodily discharge, like mainly feces, nasal oral discharge if possible. And these viruses are really capable to attach to farm equipment, pets, like manure and other surfaces. And with a high virus load on farm, it can infect like the subsequent flocks. So review biosecurity, pay attention to all details. This is an actual case that I had a couple years ago with a recurrent IBH problem. And farm visits, like reveal some deficiency in manure handling and storage. So this is the manure pile from the last cycle or previous cycles, like multiple cycle, and their contaminated manure was like feces, like dead birds as well, which is only 5 meters away from the door to the barn, with some like water puddles like outside you. So with chick delivery workers, like trucks and wind direction, pest activity, very likely like these contaminated manure and also the virus particles can be carried back to the farm and infect the subsequent cycle. So manure handling, the best practice, if you can do it, is to remove it from the farm as soon as the birds are shipped. If not possible, at least have some protocols there where the manures are being properly covered and store them as far away from your chicken flock as possible. Water system. Water system, definitely, it's another spot that sometimes we get overlooked, especially on water filters. If you're having like water filters and it hasn't been changed like for a year, for a couple of cycles, this is a time like to revisit. This is an area definitely we have like seen cases where IBH virus, like adenovirus, like have been isolated. Bursal disease virus, like can be isolated. It should be changed like regularly. My suggestions like as often as possible, ideally after every cycle. And if you don't remember anything about like water cleanliness, I highly suggest that you go back to our BC Poultry Symposium. Mr. Jesse McCoy from Neogen has a really in-depth talk about water quality and water cleanliness in broiler farms in particular. Pets. This is always my favorite. We get like lots of samples, like sometimes that comes with darkling beetles like burrowing on the carcass, mites, feces like from rodents, like flies. They can definitely carry viruses and other pathogens. So do your routine insecticide treatment. Make sure you follow the label very well to use the proper concentration and rotate among different classes of insecticides. Do a routine check. And of course, don't forget all the other on-farm equipment and also services that can be harboring viruses and bacteria. Like mortality bucket, this is one common one that I found that has been overlooked like in many of the farms. Like make sure you clean out your mortality buckets like really well. So what happened in the event of an IBH outbreak? What if you have an IBH outbreak, and how do we minimize the impact? It's a viral disease. So even if you treat with antibiotics, unlikely it will help with the situation. You can consider like treatment if there are other secondary diseases like present. Like for example, you see lesions like with E. coli, like bacterial septicemia. Trying to address that hypoglycemia stage can sometimes help, like adding multivitamins, electrolytes in the drinking water. And very importantly, walk through the barns like more often to remove like the mortality and also the sick birds. And also with more human activity, there's like encourage like the birds to move around and eat. Submit samples to confirm diagnosis. As I mentioned, it's very important for disease surveillance. And with any kind of disease challenge, I would suggest everyone to review the case retrospectively to help identify red flags. Like what happened 5 days to 7 days before that break in the IBH? Like, was there a ventilation failure? Was there a computer system failure? Was the light out? Like, was the feed was there any feed interruption? Those are the things that we can review. It's not going to help the birds in this cycle, but it's a very valuable lessons that we can learn to prevent IBH in the subsequent cycles. And of course, like anaphylaxis serology is always helpful to rule out any kind of underlying diseases. So a quick summary with the breeder levels, like with good vaccination program, also with good like vaccine techniques as well. Make sure birds are not being missed like during the vaccination process. Good incubations and also chick quality are very important too. And at the broiler level, good biosecurity, proper C&D, control all your immunosuppressive factors and also the viruses. Sounds like a lot of work, but the good thing is when you are working on the practices that are involved in the control of IBH, you are also helping to prevent many other disease challenges. This is the end of my presentation. Thank you very much. And Dr. Davor and myself are happy to take any questions. Thank you. Do we have any questions? There's a Q&A section. I think I'm going to start with the first one. Dr. Ali from Pakistan. Wow, welcome. How to cope with immunosuppression occurred due to IBH as other diseases also intervene, which causes heavy mortality, such as chikungunya virus and infectious bursal disease. I think you asked this question before my presentation, so I hope I answer your question in terms of like immunosuppressive like diseases. Feel free to shoot me a follow-up question if you have one. Second question we have is there specific vitamins or electrolytes that you recommend to help? I generally don't have any preference in terms of like what brand or what vitamins or electrolytes, like pick any vitamins or electrolytes that are for poultry. One thing that I do like to pay attention would be the sodium content of the vitamins and of the electrolytes. Like make sure to pick one that it's not like ultimately having like a high sodium chloride content, because that can upset like the salt like imbalance, especially when chicks are really young. So my short answer is there's not a particular one that I would recommend. You can pick any one that are available on the market. Yeah. What type of disinfectants are most effective against adenovirus? Davor, have you came across any study that talk about like the particular question in terms of like type of disinfectants?
Well, in the lab, we typically use bleach because not just because it kills the virus, but because it destroys viral nucleic acid. So with bleach, we clarify pretty much everything and prevent any contamination. Virkon works as well in terms of what we used before. It just Virkon doesn't destroy the nucleic acid that well. So I'm assuming in the field that bleach is not really used that much. But in terms of anything else, adenovirus 30 viruses, they can survive quite well. And I remember when I was doing my graduate studies, I kept them for one little bottle of adeno I kept for 4 years, and even 4 years later in the fridge, the virus was still alive.
Wow.
It lost some titer, of course, but 4 years later, in the fridge, it didn't destroy infectivity completely. And the other thing is important to remember is that loads of virus in those infected livers are sky high. I mean, even if you kill 99.999% of the virus, there's still going to be millions of viral particles left. So really, it's kind of hard to get rid of adeno once it's seeded somewhere. Lab is different because, you know, we do work in biosafety cabinets, and, you know, we use gloves, and but, you know, in the field where we have manure, we have all the organic material, I can imagine can be pretty hard.
Thank you. Yeah, I think I can add a little bit in terms of like the cleaning and disinfections. Lots of people focus more on like, you know, the type of disinfectants, but keep in mind that like there are a couple steps that are very, very valuable in terms of your C&D protocol. Would be make sure that you remove like all the organic matters like before you apply any detergent or any disinfectants, because like if the feces, like if the manure, like feathers, like dead bodies are not being removed like from that contaminated environment, regardless what kind of detergent and what kind of disinfectants that you use, you're going to dilute it. It's not going to be effective. So make sure you have that slight step put in really well. Drying process in between like the disinfectants like being applied, so it's very important too, so that you're not diluting like disinfectants.
I think also, just to add to what you said, you're absolutely right, because from that broiler surveillance study, at processing, again, we found that 96.4% of flocks were positive or exposed to adenovirus during their lifetime. So that would tell you that you can get rid of it, but you need to decrease the load as much as you can and get rid of organics first, and then applying disinfectants and detergents can help you do that. So what you just said is absolutely correct.
Thank you, Davor. And next question is, have you seen any correlation with IVH breaking and feed changes? This question is, I need to clarify this question a little bit. I don't think I have ever seen a case where there's a correlation with IVH break and the time of the feed change or a particular ration change. But I have seen cases where when during like feed change, like for example, if there's like an error in, not an error, a delay, like for example, in feed delivery, or like feed getting kicky at the bottom of your feed bin and the feed are not kicking in properly, that generates like a period of physical interruption with the feed, but not in particular. I haven't seen a case with particularly like a feed change like that's very obvious like yet. Next question is, with pale looking chicks at day 1, is there a recommendation on what additional management practices we can do? So going back to the whole platinum brooding concept, like we can get chicks that are having like marginal like yolk infection or like omphalitis, or chicks that are not that are underweight as well, but with good care, what we call like a good brooding practice, like these chicks can survive and can make it as a really good like size birds and healthy birds at the end of the cycle. So all my recommendations would be a very long list of like the platinum brooding checklist, like make sure that they get to like the feed like right away, check their crops like off the feed of the chick, like make sure after 24 hours like they have a good crop filled. That's an indication that they're eating, like they're drinking, air quality. It's also, this is a question is also like quite seasonal too, right? If it's in the winter, you look more for poor ventilation, like high carbon dioxide, like in the barns. In the summer, I look more for like factors like overheating and things like that. So I do suggest you, if you haven't taken a platinum brooding class yet, like that's a great place to sign up, and or you can email me. I'm happy to share that platinum checklist with you regarding all the steps that all the management practice that to focus on for day-old chicks. Any other questions? Okay. Let's see. Next one is, upon a confirmed case of IVH at cycle end, does it help to heat treat the manure before cleaning out the barn? That's a very good practice, like in general. Lots of the viruses are sensitive to heat. Like, however, when we are talking about the heat, we have to be very specific about like temperature and also the duration too. If you're heating the barn, like for example, at just like 50°F for a couple of hours, it's probably not going to work versus like 100°F for 100 hours. Davor, do you have any heat information, sensitivity information in terms of the adenovirus? I saw one source with, it was like 56°F for 4 hours, and the virus like still survive.
Well, yeah, I don't have any specific. I can find lab control stuff and share, but it will be influenced as also with the amount of organic material. So you know, more heat for longer, you know, will be needed, you know, for places where you have lots of manure, organic stuff and whatnot, versus you know, less heat for less. Certainly, at some point you can kill the virus completely with heat, but how practical this is, I honestly can't say in field conditions, because I'm not the one who goes to farms.
I can speak in the case of like, for example, IoT. The IoT virus would be sensitive to manure heat treatment for 100 degree Fahrenheit for 100 hours. So with that, such a long period of time, not only that you kill lots of viruses, but also like other bacteria and also like cocci too. So in general, it's a good practice like to heat treat the barn. However, really have to take into consideration like of the time, temperature, and also it can be very costly too, right? Like heating up depending on how big the barn is.
I generally speaking think, and just may add, is what any of those treatments does will not completely eliminate the virus. So what you're trying to do is you're trying to decrease the load, because if birds come from breeder flocks that provided them with some solid level of antibody, what you want to do is you want to kind of allow those birds to, well, because they're going to slowly lose their maternal antibody by 3 weeks, to get exposed to whatever is in the barn at the low level, so can develop their own antibody responses before they get sick or without getting sick. When for cases with vertical transmission, nothing's going to really help. Like you can have 100% clean barn. If virus comes with birds, that's it, end of game.
Thank you. Any other questions? Okay. If we don't have any other questions, I would like to say thank you again, Dr. Ojkic, for giving us like your time like very generously to share your expertise in terms of adenovirus like prevention. And for those of you who have like additional questions, like feel free to email me. And would like to remind everyone, next section next month on July 21st, our next BCPS would be talking about embryology 101 and how it is and the importance of it in commercial poultry production. We're very lucky to have Dr. Keith Branwell as our presenter from James Way.
Well, thank you for inviting me, and greetings from Ontario, and we'll meet hopefully at some point soon in person again.
Absolutely. Hopefully in person soon, and then we'll have to get comfortable with presenting with some audience again versus like a computer screen. Okay. Thank you very much, everyone. Have a good day.